So we can potentially adapt, because your genes, you cannot change them. If you're born with a set of genes, that's it. You don't change. But your epigenome can be modified by the nutrition you have, for example, from birth to weaning. So the amount of milk, the type of milk, real milk, powdered milk, all this is capable of modulating the consequences.
Hello and welcome back to the Dairy Podcast Show. My name is Barry Bradford from Michigan State University. Today I'm honored to welcome Professor Marc André Sirard to the show. Professor Sirard is the Canadian Research Chair in Genomics and Reproduction at the University of Laval and the Director of the Reproduction Research Center there. He completed his DVM from the University of Montreal and then a PhD at the University of Laval. And after spending a year at the University of Wisconsin, then joined the faculty at the University of Laval in 1987, where he's been working ever since. So Dr. Sirard, thank you for joining us and welcome to the show.
I'm very happy to be here this morning.
So I'm looking forward to this conversation. You've really been, I'd say, right in the thick of things during a window of huge developments in reproductive technologies, I would say. Can you tell me a little bit about how you got interested in this? Were you from an ag background? How did you get drawn into this area?
Yeah, well, I was interested to be a veterinarian because of my childhood environment, and I was like a Doolittle vet. But when I went to vet school, I realized that rather work with large animals than dogs and cats. I thought it was more purposeful and useful. And when I did my vet degree, it was the time where in vitro fertilization started in human in 1978 with Louise Brown, but in bovine, the first IVF calf was made in 1981 in Virginia, and some of my professors participated to that. So as a new vet, I said, oh, you go to practice because you're not really a vet until you have hit the road and practice yourself. But I wanted to do something a bit more specialized. And in dairy cows, reproduction is kind of the big topic to explore. And that's what made me decide to try to do IVF in Canada in cows in the same lab where they were doing human IVF. And they had decided to use the cow as a model. That goes back to 1982 when I started there.
rYeast40 is a natural biotechnology from ICC designed to boost the health and productivity of animals under challenging production systems. rYeast40 performance is supported by an unique processing technology that results in a pure product containing high levels of beta-glucans, MOS, and yeast metabolites. These factors, combined, promote the ruminal and intestinal modulation, helping the animals to reach their full potential.
Can you talk me through a few of the contributions you've made? I know there was 1,000 little steps in making IVF, pretty efficient at generating calves. How did you participate in those developments?
Yeah, the first calf that was born in the U.S., they used a surgery to cut the cow open and get the ovary out and aspirate the oocyte. So that was not very healthy for future reproductive capacity of that animal. So we copied the human by using a laparoscopic approach. Laparoscope is a long tube that you insert in the cow belly, so you don't have to put them under. In fact, you can do that with the cow standing. So we did that, and that was working okay. But we have, like most people around the world, we couldn't get fertilization of the oocytes. And at the same time, that was funny. I had a problem with my long laparoscope because the cow has a big belly. So the laparoscope was like a meter long. So had coagulation. The blood was coagulating in the tube. So to prevent that, I created a medium with heparin. Heparin is an anticoagulant. So adding heparin in large amount to my flushing media was preventing coagulation. But at the same time, as I discovered when I went the year later to Wisconsin that heparin is the factor that make bovine sperm fertile. Surprisingly, but I didn't know that until I was in US and working with those guys. So it was kind of by luck that laparoscope coagulation created a context where we had the right product at the right time to capacitate the sperm from bull that was coming from fresh bull and fresh semen from bulls. And then it started to work, and we make the second IVF in the world, the third. So we made 9 of them during my PhD. So that was kind of, okay, now it works for this species. And we have a process. So a lot of people starting to copy that. And I've teach laparoscopy in Texas and Colorado, different places. And that became kind of the way to treat infertile cows. But there were not that many. And that has really evolved into not an infertility treatment as it was at the beginning, but as a treatment to multiply genetics as it is today. So there's been a lot of changes occurring over the years that changed the purposes. While in human, it's still to treat fertility. So though, let's say those technological development have moved in different direction because of the different perspective in animal selection and fertility problem we have to deal with.
Yeah, you're right. And part of that has been, I guess, like any technology really, is as people got good at this, the cost, for a commercial dairy producer, the cost of doing this has dropped and dropped and dropped. And there's one AI company in the US that will give you 100% beef embryo to put in a cow for a shockingly low number. And I can imagine that you having been involved all the way through that process, that's been a really interesting development to watch.
In Wisconsin, what we did in Neil First Lab in Madison, we kind of optimized the use of oocytes we were getting from slaughtered cows. So the cow goes to slaughter. Normally, the ovaries go to the bin, and we collect those ovaries right after the animal is processed, within like a few minutes. And then we can use the oocytes that are in follicles in that ovary to produce embryos. So first we mature the oocytes, like takes 1 day. Then we put sperm, takes 1 day. Then we put them in a special good media for them. It takes 1 week. And then we can put them back in cows. So that generated a huge amount of embryo, cheap embryo, as you kind of say. So we could develop all sorts of technology like sexing, cloning, and all this because we had the material very accessible in this species. That made us learn way more than they could in human because of the access to the material. So the bovine has been a model for human for forever because of that.
So give us an update on, in your interactions, with the industry in North America. Where do you see some interesting uses of IVF for herd management today?
Well, it's been mainly developed for the high, genetic value, mainly Holstein. It was trying to reduce generation interval. So using not infertile cows, but Hereford to give a lot of embryos, and therefore within 60 days, you can have 3 oocytes recovery and get like 60 embryos. You can do the genomic analysis on them. You can sex them, and suddenly you have kind of a choice of embryo to implant. So it's really powerful for genetic progress, and as it has been used in Moet in small herds that the AI company have for increasing or accelerating genetic gain. So of course, there's been used to make bulls from those very young Herefords using even young bull at the same time. So combining those 2 teenagers together, you can go very quickly to the next generation. And now, not in the male, we cannot go prepubertal. We don't have semen. But in the female, now we can go 5-6 months of age to start collecting oocytes by superovulation. Although it's a bit controversial because people don't like to realized that we can use younger than puber animal, using non-surgical approach still, but there's some pushback in terms of how far should we go in terms of using younger and younger animals for produce those stub bulls.
I know, I think growing out of that area of research and your interests, you started to get into a related field called epigenetics. Can you first explain to us what that term even means?
Yeah, so it's kind of a new kid on the block. Epigenetics means on top of. So it's information that is added to your DNA sequence that has value for, let's say, controlling what the genes are doing. Because in our body, we have like 200 type of cells. They all have the same genome. The skin and the liver they have the same genome, but they don't use the same part of it. So in our genome, we have like 25, 30,000 genes in human, or cow is the same. So in each cell, we use like 12,000 of them. But in the skin, for example, we have pink keratin, and in your eye is transparent keratin. So the genes of pink keratin is turned off in your eyes, and the transparent one is turned off in your skin, and vice versa. So it allows all sorts of cells to have different function, even if they have the same genome. And the way it's done, genome is like a book, and there's sentences with instruction. And when we put methylation on the letters, it's like having a pen writing on it, so you don't see the letters anymore. It's carbon. It's methylation. It's really a carbon, so it's really like a pencil on top of it. And then this genes like the pink keratin cannot be read in your eyes, so that gene is silent. So this is kind of the basic concept of epigenetics. But now we realize that this programming of cells for their own fate is sensitive to the environment. So as we grow into an embryo and then a fetus, and the same from human or cows during pregnancy, the amount of food will have an impact on the size of the baby, the future health of the baby. If the mother is smoking or is drinking alcohol, now we have a lot of evidence that the health of the infant out of it will be impacted. That's the same in cows, but what we're looking at, because our cows are not smoking or drinking alcohol, they're submitted to metabolic stresses. And those metabolic stresses is probably the only way that our genome is sensitive or responding to, because the genome of cows and human have been, evolving for millions of years. So to change something takes time. To change a gene, have a mutation and have this mutation selected, it takes hundreds of years or hundreds of generations. But with the epigenetic adaptability, it's like we have some plasticity in our working genome. And if, for example, during the early pregnancy, there's not enough food, and the animal is starving because there's a drought or whatever, there's this seasonal climate change, for example, and the calf coming out of that cow will be much more efficient to accumulate energy, but will live longer and it will have a reduced fertility, which makes sense in a longer perspective. If you want your species to survive, well, you create smaller animals, don't reproduce too much, and are more efficient to extract energy just to do basic survival, not to make milk. So the concept is there. The problem is, in dairy cow, we're trying to breed our cows when they're losing weight because they're at the peak of their lactation. So doing that, we have created similar context as, let's say, obesity in humans in everywhere in the world. I would say in the United States, but now it's worldwide that there's an epidemic of obesity, and that is also epigenetic because there's no gene for obesity. So this is the effect of the environment in our system. So in cows, we don't have an obesity problem. We have an infertility problem that is growing because of this metabolic programming that is going kind of across generation and sometimes more than one generation. So the selection for much higher production in the last 40 years has been associated with the reduction in fertility, and all the efforts of the AI company in our group to select a better gene for fertility has been not so successful because it's not really genetically derived or driven. It's the amount of food that controls our reproduction, and the fact that we're breeding cows when they're lacking food because they're losing weight, because they're making 10,000 kilos of milk a year. So that creates a pseudo or a fake starvation conditions to which the embryo adapts. And the next generation, the embryo is more feed efficient. That's good for us because we don't have obese cow. They give a bit more milk, which is kind of surprising. But the cost to pay is fertility. So that's why I have a lot of work to do for the rest of my career, certainly, because fertility problem is not going away. It's increasing because the metabolic programming of the cow has been so well optimized for milk production. So that's kind of a summary of what is going on and what we're trying to understand. What's the molecular process of having this DNA methylation during early embryo that results in genes that behave differently later in life, so we can potentially adapt, because your genes, you cannot change them. If you're born with a set of genes, that's it. You don't change. But your epigenome can be modified by the nutrition you have, for example, from birth to weaning. So the amount of milk, the type of milk, real milk, powdered milk, all this is capable of modulating the consequences. In human, for example, breastfeeding is truly important to reduce obesity compared to bottle feeding in child that have been, for example, been born to mother that have slightly higher BMI, so that's kind of predisposition, and then if they have breastfeeding, it's better than if you don't give them sugar, fruit juice with sugar. It's another type of an intervention that reduce the likelihood of having a metabolic problem later on. So in cows, we would like to kind of do the same to modulate our early feeding to optimize, let's say, the later life, which is milk production and fertility at the same time.
I really like how you explain the complicated concept of epigenetics. So, using that analogy of DNA methylation is essentially a pencil scribbling over the DNA. Like, I think maybe it's a difficult question, but I'd like to see you wrestle with it. So clearly, your cells in your eye can have different scribble marks than the cells in your skin. So clearly, it can change during development with different cell types, right? Differentiating. On the other hand, you're saying that developing embryo is going to receive scribble marks that are going to influence the whole life of that animal. So how do we make those things fit together? Clearly, there can be some rewriting and some is carried over.
We're actually making, I think we're the first in the world, making a complete epigenetic map of the bovine genome using blood. And what we see is about 95% of the methylation is set by our genes, and what we call ontology is the cells that we start with one cell that divide in two cells. And for the first week of life, it goes like to 100 cells. And then there's two types of cells that comes out of it. One will become the placenta. The other one will become the embryo. So right there, you have two different fate of cells. So the enzyme that will change the methylation to change that fate, or let's say programmed by the genome of the embryo. So 95% of our genome is fixed. So everybody has, clear keratin in their eyes. It doesn't happen that we have people that are born blind. So that part is fixed. Even if it's writing on it, the process of writing on specific gene is so well established during the last millions of years that it cannot be changed, and that's okay. That's ontology. That's our development from one cells to 200 different tissues. What can be changed is what was left open for change, because it has a value, is adaptation to amount of energy, food. More food, you have larger life, you have more babies. Less food, you're cheaper, and you have less babies. So it's as simple as that, but that's only 5% of the writing that is sensitive to what's happening outside. And the embryo at 4-cell stage, and we have shown that, sense the environment. It has no noise, he has no eyes, but he has chemical capacity in his mitochondria and everything to say how much energy is it out there. And surprisingly, when the embryo or the ovary, for the same reason, see fatty acid in the blood, because that fatty acid goes everywhere, the reflex is, fatty acid means we're starving. Because normally when we have food, we have glucose, and fatty acid remains low and is balanced and goes to storage and everything. But when glucose goes down and fatty acid goes up, it means that we're starving because we're using the reserve we have. We're using our fat reserve that are being used for energy. So the embryo and ovary say, wow, we're starving. There's a problem. But, for example, in human, with the lifestyle we have today, we have fatty acid in our blood all the time because we eat too much. And worse than that, we have another sugar called fructose. This tells us fructose, it's like fruit. Fruit means fall. Fall means before winter. So our liver tends to use fructose. Fructose, we should store that for later. So when we eat fructose, like fruit juice, and we store it, then the glucose level go down. And if we eat fat, the fat goes up. So our tissue, our ovaries and embryo thinks we're starving while we're fattening, which is the opposite. And across generation, we have created a whole world problem of obesity because of that. It's a bit worse in US because we put the sugar we put in pops is not sugar cane like in Europe, is corn syrup, which is more fructose. So we've been programming in America our people to become, let's say, diabetes type 2 more and more because of that process of. So the same mechanism works in cows. So we just have to learn exactly how it works so we can better define, because in human, we don't control the environment. But in cows, we do control what they eat. We control everything. So it's a very good model to understand how the environment, the food environment, can program later health, because those cows, if they stay in the herd 1 or 2 more years, that's where, I'd say, the bacon is, that's where the money is keeping those cows up to 6 years, which is not what's happening these days because they just go infertile or mastitis. So we can better program these animals early on for reducing those problems, keeping the same milk production. Suddenly, the profit is going up. The greenhouse going down because we raise less earthers. So there's a lot of pluses of maintaining cows 1 or 2 more years in the herds and raising less cows per year, because if you keep them, you don't need that many. So that equation now is key for dairy producers to optimize their economical gain out of the process. So science is at the service of this. We just have to keep describing it and understanding it. So it's in our management system, and then we can get a lot better in making cows live longer.
The point you make about the environment that the early embryo is in, I mean, that leads pretty quickly to questions of, has that been tested in IVF scenarios, or have you sort of tried to compare epigenetic responses in IVF calves versus natural?
Yes, I mean, there's only one study before we did that in Quebec was from Pete Denson, University of Florida, was looking in one herd IVF versus embryo transfer versus AI. Why we have to compare IVF with embryo transfer? It's because for both we use a recipient and it's a mother, and the mother has an impact on the health of the future baby. So if we have a cow that is milking and pregnant, you cannot compare it to an heifer bearing an IVF embryo. First of all, the heifer is still a teenager, so even in metabolism, that gives a different impact, which is not always good to have an heifer as a recipient because of that. So what we did in Quebec, we have a kind of big co-op system. So we have data sets since 10 years old from all the robots, everything. Also the farms, we have a milk recording system that is across the state and very complete, most people. And then we extracted the IVF animal out of that with the collaboration of CMEX Bovitech, who allowed us to know, okay, which are the cow in the field that are coming from IVF. And we were able to compare like close to 1,000 animal IVF versus, and we had 15,000 ET and like 300,000 AI. And this is in herds that we compare herds that had we compare herds that had IVF cows in them. We did not compare herds that have no IVF because management could be different. So we tried to really compare apples with apple here. And what we found is the IVF animal, we compared to the first 3 lactation, 1, 2, 3, they had a bit more milk than the regular ET cows or AI. So that's a good news. But we normalize this gain in terms of genetic value because we have the genotype of both parents, and we have the predicted value of milk, and the predicted value of milk was not as good as it should have been. Although because for IVF we use the elites, of course they're making more milk. This is the elite. When you correct for this genetic, let's say, trend or value, then it's not significant anymore. They're not making more, they're not making less, but they're not making as much as we would have hoped them to do. So that's for milk. For fertility, we also have like 1 point out of 15 that is lower score in daughter fertility. So we can, let's say, if I use a crude number, those cows takes an average 5 more days to get pregnant. Well, you think that's not much, but every 4 cows, it's a full cycle that is not fertile. Now we use injection and all this to minimize the effect of this lower fertility. But there's a catch-22 problem with that. When you use drug to synchronize a cow, and then you report that this cow is pregnant, oh, she's have a good fertility. No. So the bull and the parents behind it have points for fertility, while they shouldn't have points for fertility because this cow needs to be injected to be cycling. So we have a corruption parameter in the system, which bothers the people doing genetics analysis because they have lost control. They don't know anymore who has the right genes for fertility because we inject the cows.
Biological traits that you're looking at in a synchronized animal are much more narrow than a fully natural pregnancy, right?
I know people now use AI to try to catch by the timing of dates if the people have used injection or not, because people are not recording formally if they use injection in our system. So they try to capture that differently. So I have the true fertility by eliminating the cows that they believe, because of the date, estrus and all this and timing, they believe that this cow was injected. So, but eventually it would be better to have the recording done properly. And now with the robot and all this, we're getting a bit better in terms of controlling all the information and without the participation of the breeder, kind of indirectly. So we have a change in the milk temperature, all of this with the movement of the animal. So we're starting to have better tools to better assess the real fertility.
I want to ask you about one more thing. I know you've done some work on seasonality of fertility. So we maybe think of sheep, for example, as like seasonal breeders, but we don't tend to think of cattle in that way. What have you learned in that respect?
That was interesting. We know that the cows has a problem to breed around 60 days, but to be sure of that, we compare ovary tissue, ovarian cells from cows at 30 days, 60, 90, or 120. 120, normally they're all okay, and they go back to, let's say, normal or Hereford type of fertility after 120. So we aspirate cells of the large follicle, so the ovary getting close to ovulation, synchronized, of course. And we ask the ovarian cell, how do you work? And we measure 25,000 genes at the same time. And we said, what is different in your 25,000 gene expression or the way your ovary works? And we thought we would find very complex and metabolic pathway and all this. But what we find was a bit surprising to us is we found that at 60 days compared to even at 30 and 90, they're better. At 60 days, the worst. 60 days, the ovary is lacking support, it needs more support from two pathways, which are kind of driven by one by vitamin A, beta carotene, the other one by vitamin D. And it's been known forever, and vets have used that injection of vitamin A, D, normally at calving to reduce milk fever, but more and more when you do embryo transfer, when superovulate the cow, a small injection of vitamin A, D seems to help. But now we have the kind of the explanation that the ovary is sensitive for that extra level of vitamin D and vitamin A. Then you think about it, vitamin A comes from fresh grass. And after a month, it's almost gone from processed grass and silage and all this. So people have to add it back, and it's expensive. And vitamin D comes from the sun. Of course, that's pretty easy. So the ovary will not make a calf if there's not enough food and no vitamin A and no vitamin D. If there's the same conditions, but if the animal is outside on pasture, suddenly the ovary says, oh, okay, we don't have food now, but we're going to have food tomorrow because we have sun and vitamin A. So that's the remnant of seasonality that we have found in the ovary, which explain a lot in terms of animal not going outside anymore, having the same food all year long. So we're not creating ideal condition for the cow at day 60. If we wait, and now it's a big trend, instead of trying to inseminate the cow at day 60 like we learned at vet school, now it's more, if the cow is making a lot of milk, we take a BHB sample. So we measure the amount of BHB is an indicator of the fat, the cow using her fat for energy. BHB is a fat byproduct. So when we use our fat cells, we have more BHB in our blood, and we can measure that with a keto stick, very cheap. And people are using that and say, okay, this cow is still positive, not acetonemia, but just positive for BHB. We're going to delay insemination 30 days. Then we save on money. Of course, the lactation is longer than 305, but who cares? For those cows who give a lot of milk. And then we save on veterinarian, we save on AI, we save also on the health of the animal, but not being pregnant that soon, because those milk machines are incredible. I mean, they produce so much milk now. So giving them a longer break seems a kind of very good way. And we just kind of confirmed with our analysis that the ovary was recovering at day 90. So that was maybe for those cows the best time to breed them, not to try to push them at day 60, but wait for their BHB to get lower. So, and then as we were talking about epigenetic, it's better for the next generation because if the cow is bred at day 90, she's not losing weight anymore. Her body scar is stabilized. And so the generational effect is minimized.
That's really interesting. I've heard people discuss sort of delaying first insemination. I haven't heard people discuss that strategy of using BHB as a tool to decide which cows to apply that to. I like that. That's an interesting idea.
It's time for our famous 3. AB Vista helps dairy producers maximize their herd potential with feed intelligence and targeted ingredients to optimize rumen function and overall animal health. From young calves to lactating dairy cows, AB Vista is here to combine industry-leading products and optimal feed strategies to increase your ROI.
All right, we better draw this to a close. Fascinating. I could talk about this for a long time with you, but we like to throw 3 questions to every guest at the end of our conversations. So my first one is, what's your favorite dairy-related book or resource?
Well, as a scientist, and especially in genomics, it's very clearly scientific papers. So I go to PubMed. I have all sorts of automatic alert now, and now I'm getting really interested in mitochondria. So I would read that at night sometimes better than do anything else, because that's what fascinates me. So that's how I get the information of what are the new trends coming up, is using the right keywords in the system.
Yep. Especially today with the open access journals. It's amazing what's available to anybody anywhere in the world through PubMed, other tools. That's great. What about your favorite book or resource outside of agriculture?
Well, what I read is normally always related to my work. And when I don't work, I go outside. I'm a nature lover, so I'm hiking and see kayaking nearby. There's whales around where I am, belugas and big whales. And we can, sometimes they come very close to us. In St. Lawrence, it's one of the only places on the planet. I mean, I know in Hawaii, I've been there. I mean, the whales come close to shore, but not as close as they come because of the topology of the fjord. So they come very close. So we can encounter, those big mammals as big as a school bus, and they make no waves when they come out. No waves at all. It's just amazing how beautiful they are. So that's what I love to do, is going outside and biking, kayaking, hiking.
So the first time you were kayaking and a whale came up next to you, did it kind of stress you out a little bit?
Well, not much, but I mean, because normally they blow air like a few minutes before coming back, so But my wife used to scream when they come closer, and we don't expect them. She's afraid, they're gonna make us, go over. But they're in their element. I mean, they know exactly what they are. So when they come close to us, because they want to have a look at us.
That's amazing. It's a good way to put you in your place, right? We're really pretty small beings here. All right, our last question. In your opinion, what sets successful dairy professionals apart from those who are less successful?
Open minds. We learn a lot of things from different places. We have to think critically, have an open mind and think critically of, there's so much information. Yes, filter it, to be open to new ideas. Like this epigenetic thing was not present 15 years ago. That's completely new, and people have not been trained at school. I'm starting to train the agronomists at our university with that. But it's only like 2 hours of epigenetics. Well, it's way more important. But also, nutritionists don't realize they've been doing epigenetics forever, because by controlling the sow's intake and looking at the piglets' output, I mean, it's directly programming metabolism of those, and so that's been around. But now, and we have to be open that it's playing a role in addition to genetics, and open our curiosity to it and say, okay, I have to learn more about it. So open-minded and critical thinking.
All right. Well, that wraps up this episode of the Dairy Podcast Show. I want to again thank Dr. Marc André Sirard for joining us with a fascinating discussion about epigenetics and in vitro fertilization and all things reproduction. Thank you.
My pleasure.
And if you haven't subscribed, please hit that button so you don't miss the next episode. And until next time, this is Barry Bradford signing off.